This video is on transport mechanism, particularly transport. So this is a classification of various transport mechanism. First one is passive transport, which includes simple division, facilitated division, aquaporins, ion channels, ionophores. This video has already been uploaded. You can visit that. Second one is active transport, which is currently being described. This include unipodes, import, antipode, exocytosis, and endocytosis. Endocytosis is again of three types: pinocytosis, phagocytosis, and receptor-mediated endocytosis. Third one are the pumps, which include sodium potassium ATPase, calcium pump. Fourth is transcytosis, and fifth is ABC fat. Coming to active transport mechanism, which can be classified as primary, secondary, active transport. The first classification: primary refers to energy is utilized directly, for example calcium pump. So when with ATP hydrolysis, calcium ions are either moved in or out of the cell. Secondary active transport, the energy is utilized indirectly. Here sodium entry into the cells requires hydrolysis of ATP, and this simultaneously pumps in glucose along with sodium. So along with sodium, amino acids are also transported. So this is secondary active transport, where energy is utilized indirectly for pumping in of glucose and amino acids along with sodium. So the next set of subclassification of active transport is unipodes, import, and antipode. In unipode, single molecule or the solute is pumped inside the cell. For example, calcium pump, which facilitates entry of calcium ion into or outside the cell. Symport, here two different solutes are pumped into the cell or outside the cell in the same direction. For example, sodium dependent glucose transport inside the cell, and sodium dependent amino acids, where the amino acids along with sodium enter into the cell. Antipode mechanism: two solutes are transported in the cell in different direction. For a good example is sodium potassium ATPase. For every 3 molecules of sodium pumped inside, 2 molecules of for every 3 molecules of sodium pumped outside, 2 molecules of potassium enter into the cell, because sodium is the main extracellular cation, and potassium is the main intracellular cation. The other good example for antipode is chloride and bicarbonate exchange occurring at the level of peripheral tissues of the cells and also at the level of lungs. The next set of subclassification of active transport is exocytosis, where under appropriate stimuli what happens is secretory vesicles bud up from the Golgi, they move towards the plasma membrane, fuse, and release the content to the cell exterior. So good example is release of insulin by the beta cells of pancreas, then release of acetylcholine at the presynaptic nerve terminal, and release of trypsin from the exocrine pancreas are examples for exocytosis. The last one is endocytosis, which is of three types: pinocytosis, which refers to cell drinking. Good example is engulfment of the nutrients. The next one is phagocytosis, which refers to cell eating, that is engulfment of the pathogens by the WBC or the leukocytes. The last one is a receptor mediated endocytosis. Example is uptake of the LDL by the target cells. Now here in receptor mediated endocytosis, the LDL binds to the clathrin coated receptors on the target cells. The LDL is internalized and released to the cell interior, and the receptor is channelized back to the plasma membrane for the uptake of the further LDL molecules. So other examples include transferrin uptake by erythrocytes and entry of poliovirus into the target tissues. So this was all about active transport. So thank you. The next video will be on ABC transporters and transcytosis.